Bismillahir Rahmanir Rahim. I am Aqib Rehman, Department of Microbiology, BS 7th semester, Cohort University of Science and Technology, Cohort. Today we will discuss the topic potential transmission source of Helicobacter pylori infection, detection of H. pylori in various environmental sample. This is the research paper which gave the concept of such topic. This research paper was published in Journal of Environmental Health Science and Engineering. These are the content of presentation: introduction, morphology, characteristics, disease and diagnosis, transmission, treatment, methodology for detection, result and discussion, research studies, and references. Introduction. The word pylori comes from the Latin word pylorus, which means gatekeeper and refer to the pyloric valve. What is meant by pyloric valve? Pyloric valve is a strong ring of smooth muscle which at the end of pyloric canal which control the outflow of gastric contents. Helicobacter pylori is a common human pathogen which infects up to 50% of the world population. Helicobacter pylori colonizes the human stomach and is responsible for peptic ulcer, lymphoma, adenocarcinoma, and stomach cancer. Helicobacter pylori infect the epithelial cell lining the stomach and must pass through the pyloric valve in order to infect the duodenum. So it means that it does not infect only stomach but also small intestines. H. pylori germ can enter into the body and live in digestive tract. After many years, they can cause sores called ulcer. Infection with H. pylori is common. About two-thirds of the world population has it in their bodies. This is the good picture of H. pylori. Morphology. If we discuss its morphology, H. pylori is spiral shaped. The bacterium can appear as a rod shaped also. Microaerophilic gram-negative bacterium they are, measuring its size approximately 3.5 microns in length, 0.5 microns in width. It is multiple unipolar flagella and it is actually motile. These are the some good picture of H. pylori, and some are the animated and some are the microscopic picture of H. pylori. Now we discuss its characteristics. And characteristic, first we discuss the taxonomic classification of H. pylori, in which it belongs to kingdom Bacteria, phylum Proteobacteria, class Epsilon Proteobacteria, order Campylobacterellus, family Helicobacteriaceae, genus and species are Helicobacter pylori. Furthermore, in characteristics, H. pylori is a gram-negative bacterium, usually spiral-shaped, while coccoid-shaped appear after prolonged in vitro culture or antibiotic treatment. These coccoids cannot be cultured in vitro or in lab because it is temporary structure of H. pylori which is gained by the treatment of antibiotic. A key feature of H. pylori is its microaerophilicity with optimum growth at oxygen level of 2 to 5%. H. pylori culture occur at 34 to 40 degrees centigrade with an optimum of 37 degrees centigrade. H. pylori is considered to be a neutrophile. Growth media. Which media is preferred for the culturing of H. pylori? H. pylori is fastidious microorganism and require complex growth media supplemented with blood or serum. commonly use solid media for routine isolation and culture of H. pylori. Columbia or Brucella agar is used for the culturing of H. pylori in which is solid state media. Liquid media usually consists of either Brucella and molar Henton. Now we talk about the disease and their diagnosis. First we discuss disease, which type of disease they cause. H. pylori cause stomach inflammation, gastritis, peptic ulcer disease, certain type of stomach cancer. Diagnosis. How we diagnose the H. pylori infection? We done certain type of test in which first is breath test, blood test, stool test, biopsy, sometime upper endoscopy, also done for the diagnosis of H. pylori infection. In diagnosis, urea breath test is used. In this test, carbon number 13 and carbon number 14 labeled urea is ingested by patient. If H. pylori is present, the urease activity generate labeled carbon dioxide that can be detected in the patient exhaled breath, just like shown in the following picture. Transmission. How H. pylori transmitted from person to person, enter into the bodies. H. pylori enter via the mouth and the stomach. It can only survive for a short time before it is killed by acid. The presence of the enzyme urease on its surface protects it from for sufficient time before penetrate the mucus layer. The spiral shaped motility and the production of phospholipases and the ammonium ion allow the helicobacter to penetrate the mucus layer very quickly. Transmission of H. pylori occur through fecal-oral route, gastric oral. This route, this transmission is common in children, and one are the most important transmission source as water. Oral to oral transmission may occur, and zoonotic transmission also occur in the transmission of H. pylori from person or other sources. Now, how we treated the H. pylori infection? Combination of two different antibiotics together with another drug that reduce your stomach acid. Some of the drugs that are used in triple therapy treatment include clarithromycin, proton pump inhibitor such as esomeprazole, pantoprazole, metronidazole, which is used for 7 to 14 days. Now, how we detect H. pylori in various environmental sample. First, we done sampling. Total of 88 samples were collected from various aquatic environment as well as anaerobically digested sewage sludge. Water sample were collected from various drinking water source including wells, spring and aqueducts. Wastewater samples were taken from influent and final effluent of 4 domestic and hospital wastewater treatment plants. Samples were also taken from 2 activated sludge treatment plants. H. pylori detection. How we detect H. pylori? PCR-based method was used for detection of H. pylori. Two types of PCR are done for the detection. First, nested PCR. What is meant by nested PCR? Nested PCR is a type of PCR in which two set of primer are used for high sensitivity. Both primer are different and unique properties. First primer bind outside to our target DNA. Second primer bind to specifically to the target site. This technique does not require any additional reagents or other chemical. They have only one extra single set of primer is sufficient for the process, just like given in the following picture. How the outer and inner primer are attached to the template DNA. Second, real-time PCR. Real-time PCR commonly used to measure gene expression. It is more sensitive than microarray. It helps to study small subset of gene, but those genes which are known, not used for unknown gene. This time PCR just like that which shown in following picture. Nested PCR at first primer set EUBAC27F and 1492R, which amplifies approximately 1420 base pairs fragment of 16S RNA gene region of bacteria was used. Primer set of HPNESF and HPNESR which target the 16S RNA gene of H. pylori which is used in this. After sequencing analysis of PCR positive product of environmental sample, it was defined that this band is not specific for H. pylori. In nested PCR, furthermore, the next stage we used species specific primer set of HP2, HP1 and HPNESF, HPNESR which amplify a 520 base pair and 139 base pair fragment of the 16S RNA gene in H. pylori. Species specific primer pair of HPYF1 and HPU11 and HPYF1, HPYF2 in the semi-nested PCR assay are used. This assay target the urea gene of H. pylori. Real-time PCR. Primer set of HPYF1, HPYF2 was used in real-time assay. PCR amplification was conducted and total volume of 25 microliter, 12.5 microliter premixes, 0.3 micrometer of each primer, 2 microliter and 1 microliter of template DNA, amplified product in the first and second step of nested PCR are done in double distilled water. A clinical isolate of H. pylori which confirmed by 16S rRNA gene sequencing was used as a positive control. For confirmation of H. pylori detection, the 16S rRNA PCR product from some samples were randomly sequenced for the confirmation of H. pylori detection. Result in discussion: fecal polluted environment environmental sample may play a role in transmission of H. pylori infection. The information about the presence of fecal bacterial pathogen such as H. pylori in wastewater sample can reflect their prevalence in human population. In this study, H. pylori was detected with the highest frequency in wastewater sample. H. pylori was detected with the lowest frequency in wastewater treatment plant, which could be related to presence of disinfectant in hospital wastewater. Due to disinfectant, H. pylori cannot be detected in the hospital wastewater treatment. It has been reported that H. pylori is more resistant than coliform bacteria. No detection of this microorganism in sludge may in part be related to the alkaline condition of sludge digester because of H. pylori is considered to be a neutral file. Increased risk of H. pylori infection with the consumption of wastewater irrigated vegetable. We detect H. pylori in drinking well water sample without the detection of fecal coliform bacteria. Therefore, absence of traditional indicator bacteria may not protect the water consumer from the exposure to H. pylori. Detection of H. pylori in wastewater sample confirmed that wastewater may act as potential source for transmission of H. pylori infection. Further research studies were needed for further studies would be necessary to determine the public health risk of H. pylori infection from consumption of wastewater irrigated vegetable. These are the references. If you have any question, then ask me in comment box. Thank you very much for your kind attention.